Hi, welcome to my poster. I'm Lingfei Wang from the Broad Institute. The title is Normalisr Inferring Single-Cell Differential and Co-Expression. So first of all, I have a companion talk and the SST single-cell cancer genomics. And it actually explains everything in more detail, and this poster is intended for more of how to call it, like demonstration of the plots and some of the results that we don't have enough space for in the talk, unfortunately. So basically, Normalisr is a normalization and inference framework to look for single-cell differential and gene co-expression with linear association testing. So I will not go through the details, but basically you start from a normalization, after which you can normalize expression and normalize covariates, from which you can use a conventional linear association testing, or in other words, Pearson correlation, for statistical tests. In this case, it integrates differential expression and co-expression, because for differential expression, X should be a binary 0-1 grouping, and for co-expression, it would be a normalized expression of another gene. By the way, the order I presented here is also slightly different from the talk due to formatting. So here, the first question to look at, a real data set, is about looking for gene regulations in a CRISPR-I single-cell RNA-seq screen. So in this case, you can have multiple guide RNAs in the pool screen, and with single-cell RNA-seq, you can sequence the guides, but also the transcriptome profiles. Therefore, you can look for gene regulations there. So in this case, we have a full-scale enhancer screen data set, and also a small-scale one. So those are both published in the same study. We inferred guide RNA gene associations in each of the data set, and we found actually a very strong agreement in terms of overlap here, in terms of odds ratio, and also in terms of the Pearson correlation of the log fold changes in both cases. Besides, all of the log fold changes stay in the same direction, so we also have the effect sizes and directions both matched. So then we could use the guide RNA gene associations to look for a trans association between guide RNA and a distant gene, and that is that a gene that is not its intended target and its transferrin start site. In this case, we found really overabundance of small p-values and the log fold changes in terms of the TSS transferrin start site targeting guide RNAs associations with distant or trans genes versus the non-targeting control, as you can see from the arrow bars of the red and green curves. This is shown in terms of log fold change and also the p-value. Therefore, we could indeed use the trans associations. We could find a significant abundance of significant trans associations between guide RNAs and distant genes. We also, these trans associations typically suggest for gene regulations through the cis regulatory effects, the intended targets of the guide RNA. But there's also another possibility of off-target effect. Actually, in this case, we found, because we have two guide RNAs tagging the same gene for each of the gene, as shown here, actually we can use this to look for the off-target effects. Here we looked for those guide RNA gene trans associations that showed up only in the stronger guide RNA, but not only showed up in the weaker guide RNA, but not in the stronger guide RNA. By stronger, I mean the guide RNA and the association between the guide RNA and its intended targets. So here we found actually the average off-target rate is about, it's over 50% in this case. However, we could account for the off-target effect by looking for the trans effects that show up for both of the guide RNAs, and ultimately we could obtain a less robust regulations, gene regulatory effects from the cis and trans associations. So the details can be found in the talk. And here we also demonstrated its Normalisr's purpose and effects on inferring gene co-expression networks on actually any general like 10x or MAR-seq single-cell RNA-seq data set. We only did it for particular, individual cell types, but we also tried it on other cell types like epithelial cells, besides the T cells we showed here, or also macrophages, for example, and they show comparable similar behavior, performance. Although we only did it for individual cell type rather than did it for the whole data set, including many different cell types. Generally, you can find that edges tend to correspond to the annotations of the gene function we put. On the left, for the CD8 positive T cells, we just did a brief annotation, just like with the whole, the bulk genes. But on the right side, for the dysfunctional T cells, we actually did annotation with specific for every gene based on their known functioning literature. Here, on the left side, we have log fold change comparing the differential expression between here in this case, H. poli, parasite, intestinal parasite infected mice versus uninfected mice. So these show the gene expression changes in the color. And on the right-hand side, so these are the dysfunctional T cells versus naive T cell differential expression effects. So generally, you tend to see, especially on the right-hand side, for more like literature validated results, that genes with the same function tend to cluster very well locally, indicating that, for example, if you have a gene with unknown function here, for example, then you'll be able to infer its function with its neighbor genes. Yeah, to summarize, Normalisr just is a framework that infers gene differential expression, co-expression, and actually performs much better, in terms of sensitivity, specificity, and SB than existing methods, although we don't show it here in the poster. Actually, you could find more details in the talk, also in our bioarchive preprint. We tried it in different scenario, differential expression, co-expression, and also in the screen setting of differential, highly multiplex differential expression, and it tends to perform well and on very interesting biology as well. In the future, because because it's linear association testing, it really has a huge potential to be extended into different scenarios, even in cohort settings, and we're looking forward to do that in the future. The package is available here on GitHub, and you should be able to find the link to our archive then. We are still preparing everything, but by the time you see this poster, it should be there. And besides, I haven't really put a lot of details here in the whole poster, but because I believe, poster is more like a personal interaction, so you're free to send me an email or what's available method during the ISMB. So just contact me, and I'm happy to explain to you to the details at every step how everything works, and also potentially where Normalisr may apply. I feel especially co-expression would be a type of new analysis would be to work on even most generic single-cell RNA-seq data sets, and be able to show interesting biology for hopefully any single-cell data sets you may have. So yeah, just look at the GitHub. If you want to try, the package would be there. And also contact me of any question you may have. And thank you for listening.